cross and then make it into the seeding chart, and then I hope they don't have to sit. Yeah, me too. Yeah.
Sorry. Okay. So let's talk about what may or may not be on the test tomorrow. Monday. Okay, number 1 says, explain how Mendel's observations of traits in peas led to his theories of heredity. So the basic thing that I want you to know here is that the traits that he looked at were really simple, right? They weren't the complex things that we talked about as far as like human hair color and skin color and eye color and that kind of stuff. They were very simple traits. They were easy to distinguish. And the other thing to remember is that the pea plants were easy to manipulate. It was easy to crossbreed those and know exactly which plant and which plant were produced, going together to produce the seed. Okay, so that's kind of the main things I want you to know there.
See, number 1, that was so quick.
So quick.
Just like that.
Just like that. Okay, now listen. Number 2 says, explain the process of a monohybrid cross of true breeding organisms and how the observed phenotypes in each generation are explained by heredity of dominant and recessive alleles.
That's kind of a...
Well, it's a mouthful, but let's break this down. Now, number 1, we need to know the difference between phenotypes and genotypes. Phenotypes, remember, are how they look. And remember, that's all Mendel could see. All he could do is breed stuff together and see what the offspring looked like. He could see phenotypes. But from the phenotypes, he was able to come up with a theory of the genotypes. So the whole dominant recessive thing of the true breeding cross, monohybrid means just one trait. This is where he took like the purple flowered plants and the white flowered plants and crossed them. And what did he get?
Purple.
All purple flowered plants, right? So that's the whole dominant thing, right? The purple was dominant to the white, so when you took white plants and purple plants, the true breeding purple plants, and you crossed them, you got nothing but purple plants. But the whole point is that white gene, that white allele, didn't go away. It was just recessive. It was being covered up, and it showed back up again when he took those purple plants from the next generation and bred them. Okay? And that's the basic Punnett square stuff that we did. You take, the genes, put them together, you look at the observed phenotypes. But from all that, we came up with these whole ideas of homozygous dominant, homozygous recessive, and heterozygous. So you definitely need to know what those things are. So what would homozygous dominant mean? Two of the dominant alleles. Yeah, we use 2 big letters to to show those, but that's what it means. It has 2 of the dominant allele. Homozygous recessive has 2 of the recessive allele, and of course heterozygous means there's 1 of each. And remember, if it is a true Mendelian trait, you can't tell the difference between a homozygous dominant and a heterozygous just by looking at it. They look the same. However, in incomplete dominance and codominance, we can tell the difference. But we need to know how those things show up differently. If it's incomplete dominance, they would mix. There would be some in-between thing. In codominance, you get both. So there, the heterozygotes look different than the homozygous dominant and the homozygous recessive. The heterozygotes look distinct because the traits are showing up differently. They're either mixed or they're both showing up at the same time. All right? Now let's say we do have complete dominance. So we've got let's go back to the purple plants. We've got a purple flowered plant, and we screwed up. We lost the label. We don't know if this is a homozygous dominant or a heterozygous. How could we do a test cross and figure it out?
Unit square. homozygous recessive. Cross it.
Right. We would take that purple plant, we would cross it with a white flowered plant because we know the white flowered plant is homozygous recessive. Okay? Now there's two things that can happen, and whichever one happens is going to tell us what the genotype is. If the purple flowered plant is homozygous dominant, what would all the offspring look like?
Purple.
They would all be purple because the dominant genes are going to crowd out those and overshadow those white genes in our homozygous recessive. When it was heterozygous, what would we expect to see?
Like half and half.
Half and half, about. Okay? I mean, even if you saw a single white flower plant, you would just know it's heterozygous, but we would expect to see on average about half and half, about 50% white and 50% purple. We can show those through those Punnett squares. So, Katie, you were not wrong in the fact that a Punnett square would come in handy there in order to figure out what the different outcomes could be. But we actually have to do the breeding thing to do the test cross. Okay, so we use the test cross when we have an organism where we don't know its genotype. All right, number 5, explain a pedigree and be able to use it to investigate genotypes. So a pedigree is basically just like a family tree, except it's made up in a way that shows traits. By shading them in, we can tell this thing has, or this person or organism. I mean, we use it on people a lot, but you could use it on rats or plants or whatever. Doesn't matter. Sir? So you can do a pedigree for a line of dogs or a line of pine trees. Just depends on what trait you're looking at. And so we're going to shade things in to show those, and what we can do is then kind of use logic to figure out what the genotypes of certain individuals are. Okay, remember, if you see a recessive individual, we know that genotype. And then by looking at how those things match up, we can kind of figure out what some of the other ones are based on how those alleles had to be passed on. So a pedigree is kind of a stand-in for a test cross in some cases. You know, you don't necessarily have to do a test cross if you know what the parents and grandparents look like. You can figure it out that way. So a test cross would be more like, oh, we don't know anything about this plant or anything about this mouse or whatever. Okay? Yes. Now on the test, I will tell you, there'll be a pedigree there. You'll have to label it correctly. So put the generations and the numbers of the people. And then you'll have to figure out genotypes for as many as you possibly can. Remember, don't guess. Don't go, oh, this one probably is this. Only fill out the ones that you know for sure. If you don't know, you can put a question mark. Or on the test, you can just leave it blank. You don't need to go write question marks on everyone you know.
So you have to label the generations like that?
Yes. You'll have to label it and then label genotypes. Okay, let's go back to number 4 just briefly. One thing I forgot to talk about there. When would test crosses maybe not be the best way to go about figuring out genotype? Why humans?
Well, we don't take a lot of offspring.
Because it takes a long time to have any offspring. Gestation is 9 months.
And you only get one.
And you get 1, maybe 2. So,
Possibly 4.
okay, yes, possibly everything. But the point is, test crosses only work really well if you have organisms that reproduce quickly and or reproduce a lot at a time. You can have a lot of offspring at once. So plants that produce lots of seeds, that's not bad, as long as it doesn't take forever to make those seeds. Okay. Okay, and those of you that come in and missed the first little bit, I have been recording this, so you'll be able to catch it later if you need to. All right, number 6 says you should be able to create Punnett squares, be able to use them to predict phenotypic ratios. That should be ratios, not rations. ratios for monohybrid and dihybrid crosses. This is the thing we're really after. Okay, now there will be probably a question on there that asks you to make a Punnett square. Kind of show that you know how to do it, but as far as the notation that you use, I'm not going to be all that picky about that. As long as you can use it to figure out the right phenotypic ratios, that's the main thing.
Is there going to be like a dihybrid one?
There will be a dihybrid one on there. Possibly it's already filled out for you and you just have to find the phenotypic ratios. That could be a thing that might happen.
Yeah, it's like the passport. That was a really big one.
Yeah, so 16 boxes, that's a dihybrid
But
cross.
it's possibly already filled out. That would be nice.
Yeah, to save time, it's possibly already filled out and all you have to do is read it and find the ratios.
Okay, now I'm...
It would be good to look over those again. Okay? All right, number 7 says explain Mendel's laws of heredity and how they relate to the process of meiosis. So, what?
Law of segregation.
Okay, law of segregation. Law of independent assortment.
Oh, because the things get separated, I think.
Right, so the chromosomes get separated, separate from each other. Remember, in one of his laws also was that there are different traits, and that one trait could be dominant to another. Now, you know that's not always true, but in Mendel's world, that's what it was, because all the traits that he looked at, it was simple dominant recessive. So he said each trait there's two different kinds that you could have. You get one from your one parent and one from the other. Okay. Remember, that all comes down to, in meiosis, the chromosomes separate, and how they flop down is completely independent of each other. One chromosome does not affect another, and there's lots of different ways that they can do that. Talked about in the last chapter. Okay, number 8 says explain sex-linked traits and how they are passed down through generations.
And only on the X.
Only on the X chromosome. Remember, a couple of things. Number 1, that means that males are not carriers. Either they've got the trait or they don't have the trait. But the other thing is, that means that males always get that trait from who? Their mother, because they get the X chromosome from her. Y chromosome comes from dad. So whether we're talking about male pattern baldness or hemophilia or color blindness, we tend to see it skip a generation because the male will pass it on to his daughter. The daughter won't show the thing because she's just a carrier, and then she'll pass it on to her son. And she may not. I mean, that's just a probability thing, but that's usually the way we see it unless something else weird happens. Okay? And remember, what's the probability that a guy that's colorblind and a female that has 2 normal genes will have a son that is colorblind? 0, because that X chromosome won't get passed on to any sons. Okay, number 9 says explain more complex heredity like polygenic traits and traits affected, that should be by the environment.
Why is it
Does it? I must have fixed that on yours. So, polygenic traits, remember, are traits that are controlled by more than one gene. And a lot of times there's like a cascade. This gene has to turn on this thing, which will turn on this thing, and will turn on this thing, and then you get a certain color hair. So polygenic, remember, poly means many, genic just means gene. Then we also talked a little bit about epigenetics and how some traits, well, we talked about a couple things. Some traits can just be directly affected by the environment. We talked about how flamingos' color is all about what they eat. Okay? We talked about how your fingerprints are not just down to your genes, also the conditions, how your arm was wrapped around itself or whatever in the womb, and that's why identical twins have different fingerprints. But then we talked about actual epigenetics and the fact that we're finding out now that the things that you do in your life can actually change your genes slightly, and that those changes can be passed on to your offspring. So, in lots of spree, you could be changing your genes and being handed down to future generations. Awesome.
So your kids could really be just
But it could be. I mean, the basic thing that we're seeing is it could affect enzymes and health. Okay. All right, we're getting there. Almost done. Multiple alleles. Don't get this confused with polygenic traits. In polygenic traits, there's a bunch of different genes, maybe even on different chromosomes, that work together to make a trait. Multiple alleles is different. In multiple alleles, you still have one gene. You got two slots. There's just a bunch of different types of alleles that could go into those slots. So instead of purple and white flowers, maybe there's a gene for purple flowers, a gene for white flowers, a gene for red flowers. You can pick two, right? And we see that when we look at blood types. We're going to talk about next. The blood types, that's an example of multiple alleles. You got the A, you got the B, you got the little I that does nothing that we call O. You only get two of those, but any combination of the two will affect what blood type you actually end up with. All right, as far as blood types go, a couple things that you'll need to be able to know for be able to do for the test. Number one, there will be at least one question, maybe two, that will deal with how blood types are passed on and who's the daddy. So, like the worksheet that we did before, based on the mother's genotype or blood type, sorry, the mother's blood type and the baby's blood type, you should be able to eliminate any respective fathers.
Or mother.
Or mother. I will put a weird one like that on. That was actually a typo that I made at one point. But I leave it on there so that we can talk about it. It is kind of an interesting case. Wait a minute. You can't be the mother.
That's the issue.
Okay, the other thing though is you should look over those different blood types and you should be able to know who can get blood from who. So, if you have A-type blood,
A or
okay, you have B-type blood, you have AB blood, AB blood. So you should be able to know those. So if I say on the test, you have B-type blood, what type of blood can you get? That's the basic question. All right. Number 12 says explain the heredity and symptoms of certain genetic disorders. I don't know. I used to have questions on there on the test that would be like, name a autosomal recessive disorder. I've kind of taken those out. I mean, I don't know, that's just like straight memorization stuff. But we should still realize kind of how the different types of disorders work. So like how are dominant disorders handed down? We talked about Huntington's disease and how it wouldn't really work if that was a disorder that you call it disease, but not disease. You can't catch disorders. That disorder would not work if it killed people before they got to reproductive age.
which one
Because
kills
dominance,
before reproductive age?
Tay-Sachs, cystic fibrosis tend to, PKU used to kind of, we don't see that quite so much anymore, but
Those are the recessive.
yeah, recessive ones can do that because they can hide in the carriers. Okay, but I mean, it'd be good to have an idea that hemophilia is one of our sex-linked traits. And what was the different thing about sickle cell anemia? It's co-dominant, yeah. I mean, yeah, that's how it works, but that was the interesting thing to me anyway, because it's about the genetics of it. It's a co-dominant trait. The heterozygotes have half round cells and half sickle cells. And we ran out of time yesterday before I could say the last thing that I was going to say, which is what's interesting is in Africa, heterozygotes actually are more likely to live because people with sickle cell anemia obviously have problems, but people without either gene that have all normal ones are actually more susceptible to malaria than people that have half sickle cells and half not. So there it's kind of weird. There's this evolutionary pressure that are making people that are heterozygotes more likely to survive and pass their genes on, and it's kind of keeping that old sickle cell thing.
So are they having more sickle cell children?
Yes.
Because
there's more people that are heterozygotes.
They want.
Well, not necessarily. You don't want people to be sickle cell, but being a heterozygote actually makes you more immune to malaria. So it's just an interesting case in that co-dominant trait. Okay, so I mean, have a vague idea of how those work, but yeah, I'm not going to ask you on the test what is an example of a sex-linked disorder or something like that. That's just memorization. I want you more to know how the genetics work. Okay?
What do you mean how the genetics work? Like if it's co-dominant?
Well, like how it's passed. If I say this is a co-dominant trait, what is that? Okay, so that's pretty much that. It is